Well, good afternoon, everyone, and welcome to the next in the series of Swift Analytical webinars. This one's entitled Connecting Cells in 3D Spheroids and Their Potential in Research and Preclinical Applications. My name is Malcolm, and I'm responsible for the business development within Swift Analytical, and I'm going to be chairing the session this afternoon. The webinar will be presented by Dr. Anna Maria Reich, who's the R&D manager at Facilitate, and they are based in Mannheim in Germany. Anna Maria is the scientific lead for the development of new products and technologies for Facilitate. And prior to joining Facilitate, Anna Maria was the postdoctoral researcher at Karlsruhe Institute of Technology at Leibniz University in Hanover. And this afternoon, Anna Maria, she's going to be talking to us about the importance of standardization in 3D models, and we'll be comparing different technologies that are currently available for the development of spheroid-based systems. As usual, there'll be the opportunity for questions using the Q&A box facility at the bottom of your screen, and we will try to answer as many questions as we can during this time. We're leaving time at the end for that. But if we can't answer your questions, then we'll do that offline as soon as possible. And the good news for you is that all attendees are eligible to receive a free evaluation sample. So if you'd like to test one following this presentation, just register your interest in the post-webinar questionnaire. But before we start the main presentation, I'd like to introduce Dr. Meb Survey, who's going to give a quick overview of Swift Analytical for those of you who are not aware of our company. Over to you, Mebs.
Thank you, Malcolm. Good afternoon, everybody, and thank you so much for taking the time to join us. I'm Meb Survey, and I'm the Managing Director for Swift Analytical. So I just wanted to take 5 minutes of your time, and also while other people are coming in, they may be a little bit late coming in to the meeting, to give you a very quick overview of Swift Analytical. So Swift Analytical was formed 10 years ago. We're based in the historic city of York, and our aim is to bring technologies to scientists and researchers in the areas of single-cell omics, high-resolution bioprinting, 3D cell culture. We also provide high-performance reagents that enable assay developments for drug discovery. So let's just begin now to introduce one of our flagship products, and this is from a partner of ours called Fluicell. What they have is a technology that enables the manipulation of microenvironments of cells or groups of cells by having a precise reagent delivery system. So on the left, you can see the device, which comprises of a control box, which controls the pneumatics. We also have a pen holder and a Android-based tablet in order to operate the microfluidics. So on the right-hand side, we've got a video which I'll quickly scroll through. And this is the biopen tip. The biopen tip is the means with which we're holding the solution. It's got a flexible end, so when it comes into contact with the surface, it doesn't break. We fill in our reagents. Up to 4 different reagents can be used, and they can be of different concentrations or different types. And the 4 other positions that we have are the waste positions. So once we clamp it into place, what we can do is we can place it on our micro manipulator and onto our sample plate. And then we can identify the specific cells of interest that we want to target. So here we've got a single cell, and we're applying short, discrete pulses of liquid to the cell of the different types of reagents. So we can continue our study to then take it on into, say, a pulse mode for groups of cells. So this is a very highly powerful device, and it has now evolved into something that's called the Biozone 6, which is being specifically targeted for drug toxicology studies. The other product within the portfolio is called the BioPixlar. On the top, we've got the same principles as the BioPen, but it's been modified to enable dispensing of single cells with high resolution. So if I run both videos, the top video is basically showing you how we form the dispensing of a liver cancer cell. And the bottom video is showing how we can surround that with fibroblasts. The key thing to remember here is that you don't need to use biogels. So the shear forces that are encountered during the process are very low. This process is really powerful for printing fragile materials such as neurons, stem cells, hepatocytes, for example. And the sorts of tissue models we can generate very quickly are shown here. So on the top, we've got a liver model, and on the bottom, we've got skin cancer model. The next product I wanted to introduce you to is something that we call the iWash. And this is a really useful tool, especially for those that are doing cell counting on a routine basis. You may have a Countess 2, a Logos, Luna, a Biorad, Nexcelom, and other cell counter within your lab. And one of the issues with this protocol or workflow is that once you've done a measurement, you throwing away 1 pound, 3 pounds, 5 pounds worth of cell counter slide when you could actually be recycling it and reusing it. So the iWash allows you to do that. And what we're doing is we're helping labs to get onto the sustainability ladder. So we're able to wash and recycle cell counter slides up to 50 times. And this has a huge economic and environmental benefit. The next product I wanted to introduce you to was the Biofluidics BioSpot. And this is a high precision, high speed device, which enables the dispensing of cells, spheroids, and organoids with very high precision and high viability. So we can dispense from picoliter to nanoliter to microliter volumes. The next is a product range that we are proud to be partnering Blurt with. It's a company that's based out of Poland, and they provide very high quality, high precision reagents and enzymes, which our customers are finding, especially in these days where there's a big need for developing COVID tests. These tests are being incorporated in a number of tests as we speak. The key benefit here is the economic benefit. It's incredibly, it's significantly cheaper than the leading brands. And for example, if we look at the 529 polymerase, that's about a third of the cost of a market leader. So the next product that I wanted to quickly introduce you to was the Cell Dynamics weight system. And basically, this is a device which is flow-based, and it allows us to simultaneously measure the weight, mass density, and size of sphere-like biological samples using gravimetric and dimensional analysis. So now what we're able to do is to have a paradigm shift from what we've been doing now to what we're able to do with this technology. So with the cell dynamic weight system, we're able to monitor events such as maturation, weight loss, density variation, and also it's proving very useful, for example, in the area of oncology, where you're trying to target specific drugs. For more information, obviously, please get in touch with us. One last slide before I pass you over to Anna Maria is we are also working with another company called Cougar Myers, and their technology enables us to generate high clusters of highly reproducible spheroids, and these are available in the SpheroPlate. So without further ado, I wanted to thank you for your time and pass you over to Anna Maria, who's going to be talking to you about the Facilitate range of products. Thank you, Anna Maria.
Okay. Okay, good afternoon from my side. First of all, thanks to Malcolm for the nice introduction. And yeah, my name is Anna Maria, and I'm the R&D manager at Facilitate. So due to the current pandemic situation, we all know how frustrating it is to stay alone or to have only contact to 1 or 2 people in our environment. And we are all tired of social distancing. And for cells, it's the same. So they want to be far away from each other or have contact just only one cell or two cells in their environment. So preferably the cells like to be in contact with each other and to form a dense network. And today I want to talk about the importance of this connection in reliable cell culture model systems for preclinical applications. So reliable model systems are urgently needed. For example, last year, around 10 million people died due to cancer. And model systems which allow to model the disease and to get new insights about the origin of the diseases can help to find new drugs and helps to decrease the number of deaths. But also the COVID-19 situation taught us the importance of fast drug development and the indispensable role of model systems to fight against infectious disease. So such model systems are used in drug development and to screen new drugs or to check the drug safety. Currently, model systems are used as standard model systems in preclinical studies, accepting the species differences. So for example, animal cells show different cell behavior and different expression profile in comparison to human cells. And this has a big impact on the predictability of the results. Further, such animal models are not usable for wide-scale drug screening. They are expensive and they need a lot of time. And another thing is that they are limited available. And according to the international regulations of the 3R ethical guidelines, so animal testing should be replaced, reduced, and refined. Implementation of in vitro systems in preclinical studies can bridge the gap between animal studies and humans. So some of the systems are still limited in predictability, but it's possible with such in vitro models to screen drugs in a wide scale. In vitro systems are cheap, so it reduces the cost and the duration time. And in vitro models can reduce animal studies and maybe someday replace the animal studies. Currently, in vitro models can be used for different applications. They can be used for disease modeling, so to understand the origin and the progression of disease on molecular and cellular level. And the results from such models can implement into further drug development. And also in this area, in vitro models play an important role to check disease-specific responses and to use them in drug screening, but also to monitor the drug bioavailability, therapeutic efficiency, and dose-limiting toxicity. Another important area in drug development is to analyze and to understand the side effects of drugs. So, for example, drug-induced liver injury are a major source of clinical attrition, precautionary warnings, or post-market withdrawal. So accordingly, there is a need for more predictive tools to assess hepatotoxicity risk and drug discovery and therapeutic efficiency. So far, there are several systems, several in vitro systems. So far, traditional 2D in vitro systems are commonly used, but these systems incompletely recapitulate the natural microenvironment. So the cells in such 2D systems connect just to one or two cells in their environment in one dimension. And therefore, 3D cell culture models have become increasingly popular. So 3D models allow physical cell-cell and cell extracellular matrix communication in each dimension. And there are several methods to culture cells in 3D. So you can embed your cells in a scaffold, which provides the extracellular matrix. So these are scaffold-based systems. But you can also culture the cells in non-scaffold-based systems, for example, in spheroid cultures. And in these culture systems, the cells show a natural shape, and it has been shown that the proliferation rate, the gene expression, and the signaling pathway activation was similar to in vivo conditions. Further, in such 3D systems, you can build up multicellular systems. And such 3D system can bridge the gap between in vitro and in vivo drug screening, and possibly decreases the use of animal models. 3D models are used in preclinical studies and drug screening. They are a key tool to understand diseases and to assess the potential efficiency of a new compound in drug discovery. So previous studies have been shown that cells in a 3D environment activate typical signaling pathways of solid tumors and cancer-specific gene expressions. So these models can be used as disease models. But also the cell function and cell behavior and morphology was similar to what is seen in vivo. In the area of drug screening, a 3D network mimics tumor drug distribution, greater resistance against chemotherapeutic drugs than in 2D systems. But still, 3D models are not standardized in the lab. So 3D models are a better predictor, but a standardization is needed. So what kind of 3D culture have the potential to be a standard assay in cell culture? If we have a look on the scaffold-based 3D culture, so this model systems resemble the in vivo condition very nicely. So they have this extracellular matrix, but the production of such scaffolds is labor intensive and expensive in preparation. Some of these models have undefined compositions like animal-derived proteins, and this has an impact on the predictability of your model. But also, the scaffold can undergo structural changes over time, and this impacts the reproducibility of your results. Spheroids, in comparison, are 3D self-arranged cell clusters. Spheroids are simple in making, they not expensive, so they're very cheap, and they provide reproducible data. Spheroids can recapitulate the tumor environment, which is a perfect model, which makes them to a perfect model in cancer research. For example, here you can see a spheroid, and this spheroid has proliferating cells on the outer layer and a hypoxic core with dead cells, with necrotic cells. So they show a heterogeneous cell population, which mimics the tumor microenvironment. Spheroids can also reproduce the mechanism of tumor initiation and spread. So spheroids are a perfect model for cancer research. But spheroids can not only be used in cancer research. The 3-dimensional spheroids for hepatic culture have emerged as a promising tool to assess mechanism of hepatotoxicity, as they demonstrate enhanced liver phenotype, metabolic activity, and stability in culture, which was not attainable with conventional 2-dimensional hepatic models. So the spheroids have a high potential to standardize the 3D culture in preclinical studies and drug screening. There are different systems available to culture such spheroids. but they differ in the suitability as a standardized system in drug development, including high-throughput screening. So, for example, you can culture your cells on biologically inert surfaces, which are cell and protein repellent, and forces the cells to aggregate and form a spheroid. And this technology is used in our bioflow plate. But you can also culture your spheroids in so-called spinner flasks, in which stirred vessels are used to prevent the cell adhesion to the surface of the plate and allowing 3D spheroid formation. Another method is the hanging drop method. In this method, the cells are seeded in small drops of medium, and in this medium, the cell forms aggregates due to the gravity forces. Another method is the cultivation with microcarrier beads. In this method, the cells adhere to the microbeads, proliferate on the surface, and form 3D structures. In spinner flasks, the cultivation in spinner flasks do not allow a high throughput analysis in cell-based assay. And also, the microcarrier beads can interfere with your cell assay in a high throughput screening. When we compare a low adhesion plate with a hanging drop system, we can see that in the low adhesion plate, the spheroids are formed and they show a round regular morphology. In comparison to that, the cells in the hanging drop systems form spheroids with irregular shape and small cell satellites. Another big disadvantage of the hanging drop method is that it's not possible to replace the media or the drug during your experiments, which is important when you want to check the response of the drug during several days. The hanging drop system is a fragile system and it's complicated to use, so it's not suitable for high throughput screening. So the hanging drop method has limited use in drug discovery. So we provide spheroid cultures, low adhesion plates. We provide two different products. First, the 96 well plates, they are ready to use and already coated. And we provide the BioFloat Flex solution, which is an inert coating and which is widely applicable for coating chips, microfluidic devices, bioreactors. Both products are animal-free and allow a faster, time-saving generation of spheroids and gives you reproducible results for a wide range of cell types. These low adhesion plates are ready to use. That means no assay establishment is needed. So you can adjust the size and the density of spheroids by the initial cell number. So here in this experiment, we see that 750 cells per well, and after 24 hours, a spheroid was formed. After seeding 6,000 cells per well, also a spheroid was formed, but it was bigger compared to the well which was seeded with 750 cells. Further, with these low adhesion plates, you can also refine your spheroid model similar to in vivo conditions. So you can use co-culture models. Here in this example, we cultured cubic cells in red and osteoblasts together. And as you can see, both cell types form one spheroid. And this is one important thing if you want to recapitulate the cellular heterogeneity which was found in solid tumors. So spheroid plates have a high potential for standardization, but they need to meet to meet several requirements. So they need to be animal-free. They need to provide the user a freedom to operate, for example, in the 96 well plate, in chips, microfluidic devices, or bioreactors. They need to be time-saving. They need to be high throughput compatible, and it should be able to work with relevant cell lines. And more important, they need to meet the 3 core requirements. So a rapid formation of spheroids. The spheroid should be round and reproducible, and the method or the system should be reliable and robust. And these 3 core requirements and the quality and the performance depends on the spheroid plate manufacturer. With bioflow plates, it's possible to or allows the rapid formation of spheroids. In this movie, you can see astrocytes, primary astrocytes, and here you can see that the astrocytes form as round, perfectly shaped spheroids within 22 hours. So it's possible to start your experiment very early after cell seeding. It saves time, and this is particularly important when results are urgently needed. Further, these ultra-low attachment plates are compatible with a broad range of cell lines, primary cells, stem cells, and allow a reliable culture with challenging cell lines. Different ultra-low attachment plates generate spheroids with different morphology, which can impact the readout of your assay. So here you can see the bioflow plates, and in the bioflow plates, spheroids were formed with a round morphology, and in 3 other benchmark products, also spheroids were formed, but they show small deposits, irregular shape, or satellite aggregates. So in comparison to other plates, our bioflow plate outperforms in the performance. Here we quantified the data and we measured the circularity of the spheroids, which is a marker for the roundness of the spheroids. So values near 1 describe a perfect circle. And spheroids in the bioflow plates showed the highest circularity in comparison to benchmark product 1 and benchmark 2, which showed a lower circularity. Furthermore, in the bioflow plates, only 1 spheroid per well was formed. And the number of the spheroid per well, as well as the morphological, have a significant impact on the readout. So it can lead to inconsistent results, which impacts the reproducibility of your assay. Another important factor for a reliable 3D system is the formation of viable spheroids, particularly in the field of toxicology studies. In this assay, we cultured a human astrocyte cell line, and we checked the viability by measurement of the ATP level in the supernatant. And you can see that the spheroids and the bioflow plate are highly viable after 3 days of culture and after 6 days of culture. In comparison to benchmark 1 and benchmark 2 showed a lower viability. We also stained the spheroids here on the right side, and we stained the spheroids with casein, it's a marker for live cells, and with PI, a marker for dead cells in red. And you can see the live cells in the bioflow plates with a small core of necrotic cells, which is normal for spheroids. happen that cells in an ultra low attachment plate aggregate but do not form a real 3D structure. For example, here in this benchmark product, But in comparison, in the biofloat plate, the spheroids are more three-dimensional and show a more highly regular shape. Some cell lines need a high portion of extracellular matrix because this leads to an upregulation of the cadherin, which leads to a denser cell-cell contact. And after adding 2.5% of Medgel, you can see that in the biofloat plate, the cells form a highly regular 3D aggregate. In the beginning, I mentioned that a standard spheroid method need also to be flexible, usable by the customers. With our second product, BioFloat Flex, it is possible to passivate different surfaces within seconds. So in this experiment, we coated a 96-well plate with BioFloat Flex for 1 second, 60 seconds, and up to 300 seconds. And afterwards, we seeded the cells into this well, and you can see that after 300 seconds of coating, of treatment of the surface, the cells are able to form a spheroid, and also after 1 second of treatment, the cells are still able to form a nice round-shaped spheroid. The BioFloat Flex solution can be used for several surfaces, for glass and plastic surfaces. In this experiment, we checked the protein absorption of different surfaces. Here in blue, these are non-coated surfaces, and you can see that the proteins are able to attach to these surfaces. After treatment with BioFloat Flex, the protein absorption decreases and no protein absorption was visible on different surfaces. And therefore, BioFloat Flex can be used for coating multi-well plates, chips, microfluidic devices, or bioreactors. It's easy and time-saving. BioFloat Flex is a robust coating. It shows mechanical stability and scratch resistance. Here in this graph, you can see the stability over media exchange. So here, these are our BioFloat plates, and in blue, a benchmark product. And we changed the media several times, up to 12 times. And what you can see is that after several washing steps in the BioFloat plates, it's still possible to generate 100% of spheroids. In comparison to that, benchmark product 1 was only able to form spheroids up to 80%. We also checked the scratch resistance. So we treated the surface very harsh. And you can see that after surface treatment of our BioFloat product, the cells are still able to form a nice spheroid. In the benchmark product 1, it was not possible. So our BioFloat Flex coated plates are more robust than benchmark 1, and they are extremely scratch resistant. So our BioFloat plates are animal-free. They provide the freedom for the users to operate in 96-well plate chips, microfluidic devices. They're time-saving and easy to use. So they allow fast generation of spheroids for fast experimental setup. They are high throughput compatible. And more important, they meet the 3 core requirements. So they allow a rapid formation of spheroids. The spheroids are round and reproducible for perfect data consistency. They're reliable and robust. So we showed a stable quality and performance of our plate. And in my last 2 slides, I just want to show you that our plates are also applicable for relevant cell lines in drug screening. So the biofloat plates are also useful for analyzing drug side effects. For example, drug-induced liver injury, which I said in the beginning is a major source for high attrition rates during clinical studies. Biofloat makes it possible to form spheroid from difficult to culture primary cell types. So here you can see after 9 days of culture in the biofloat plate, you can see a round-shaped spheroid. In comparison to the benchmark products, which show an irregular shape and cell satellites. So the biofloat system made it possible to form spheroids on difficult primary cells. We also measured the activity of cytochrome P450 enzymes because these enzymes are necessary for the metabolism of drugs. So knowledge of drugs metabolized by these enzymes or inhibiting drugs can minimize the possibility of side effects. Being a substrate of such enzymes can be positively associated with clinical incidence of the drug-induced liver injury. And what you can see is that in our plates, the cells express this enzyme here in blue without adding an inducer cocktail. If we add an inducer cocktail, the expression of the enzyme increases in the biofloat product, but also in the benchmark products. So all spheroids demonstrate a similar robust basal enzyme activity. So in conclusion, spheroids formed in low adhesion plates are reliable and predictable model systems for cancer research, stem cell research, and drug testing. Spheroid models are applicable as standard method in preclinical studies, drug development, and support the 3 Rs in animal experimentation: reduction, refine, and replace of animal experiments for fast drug development. And the bioflow technology connects the 3 core requirements, so rapid, reliable, and round and reproducible, with the usability, and can be used as a standard method in small research labs, but also in industry. And with this, I want to thank for your attention. And as I said in the beginning, that networking matters, which is also true for science community. I would be happy to stay in contact with you. And if we can help you with your spheroid culture, just contact us, me, or MapServe from Swift Analytical.
Great, thanks, many thanks, Anna Maria. That was some interesting comparisons there, and it's interesting. Lots of questions have now been coming in, so we have quite a few to get through. First one is, how are spheroids different from organoids?
So in organoids are from patients, directly got from patients. So there are not only one cell line, it's more like a small tissue. So you can get spheroids from patients directly, or you differentiate the iPSCs to organoids. That's the main difference. But it's also possible when you differentiate iPSCs into organoids to culture the organoid in our plates.
Brilliant. From Francesco asks, when we generally coat plates with agarose to prevent cell adhesion, some agarose may be present in suspension, creating undesired debris. Does your coating solve this issue?
Yes, So we saw that with our coating, it's possible to generate one spheroid without aggregates. But if this is not possible, which I don't know, which maybe happens for primary cells or hard to culture cells, you can also centrifuge the plate, and that improves the spheroid formation.
Mm-hmm. Would you still need to add growth factors to your culture when using BioFloat, or does the BioFloat kit come with growth factors and media?
Mm-hmm. So it depends on your cell line. So some cell line needs growth factors, the addition of growth factors, but the growth factors do not interact with the BioFloat surface. So because it's a protein repellent surface, the growth factors will be in the medium and do not adhere to the surface.
Mm-hmm. Joanna asks, is it a common classification to define spheroids as not scaffold-based? If substances are added to enhance spheroid formation, like shown with the Matrigel, is it then scaffold-based?
Mm-hmm. So scaffold-based means adding extracellular, it could be a polymer or proteins which are which forms or which interacts with the cells. So, and spheroids do not have a component which interacts with the cells. I would say. Yeah.
Mm-hmm. Another question that should come in. Do you have 384 plates, well plates?
So we produced 384 plates, and they are now under quality control, and we launched the product in the next 2 weeks. So when someone is interested in this kind of well plates, we can provide a sample.
Right, that's a good timed question.
Yeah.
Pia, she's asking, how does BioFloat Flex compares to Matrigel? Is it as temperature sensitive as Matrigel with regards to being in liquid or gel form?
So BioFloat Flex is not you cannot not compare BioFloat Flex with Matrigel, because Matrigel is a 3D environment which can interact directly with the cells by contacts. So the cells start to attach to the matrigel via the integrins and then proliferate. And BioFloat is a protein and cell repellent surface, so the cells do not interact directly with BioFloat.
Mm-hmm. Thank you for that. And then lots of questions about BioFloat Flex coming in. Is the BioFlex coating efficient to hold the shape of a 3D bioprinted model in a trans well?
So I need more information for this question. So I assume the customer wants to print the cells to a BioFloat Flex surface. I'm not sure. So as I said, that BioFloat Flex is a cell repellent surface. It's not possible to print a structure on the surface with BioFloat Flex.
All right. But then for Chris Wynn, who hopefully is still listening, then we'll contact you directly, if that's
perfect.
Okay,
yes.
and discuss the application in more detail.
Sure.
Do you have a distributor in Turkey? Because someone would like to try the product.
We do not have a distributor, but you can just contact me and we will send you a sample. It's not a problem.
Yeah. And as I mentioned in the post webinar survey, there's the opportunity there to fill in the details and get your free sample. So that would be great. Can you collect the organoids from your plates for different subsequent applications?
It's possible. So when the spheroids are very big, I would say around 300 to to 400 microns, you can get the spheroids out of the well with a pipette and transfer to another well. But it depends from cell line to cell line. So some cell lines form a dense network, and some cell lines do not form that dense network. So it could happen that the spheroid disrupts during the transfer method.
Again, another case of try it and see, really. Test application. Yeah. Can the flex be used to coat other materials, such as organ on a chip?
Yes. So you can just let me show you one slide here. So you can coat different surfaces. So chips which are from polystyrene or topaz or PVDF. So it's not a problem. But maybe we can just, you can write me an email and describe the microfluidic device, and then I can give more informations.
Yeah, I think that is the message for all the attendees this afternoon. If you've got any more specific, so the offer is there to try the systems, but any more specific applications, then certainly we'd like to talk to you and just work it through. Because there are lots of different applications, as we know. Can BioFloat Flex make the cells remain adhered on the walls of the cell culture plates?
So yeah, I think when you treat your wells with BioFloat Flex, also the walls will be protein and cell repellent, so it's not possible for the cells to adhere to the walls over 96 well plates.
Yeah. How does BioFloat perform with organoid cultures?
So one of our customers used the BioFloat Flex with iPSC-derived organoids, and it works very well. So just give it a try. As I said before, organoids sometimes a little bit complicated, especially when you work with patient-derived organoids. But yeah, I would just give it a try. I would say it works, but maybe it needs addition of extracellular matrix proteins.
Venkatesh asks, how much time does it take to standardize?
So it depends on your cell line, but when you, or do you mean to standardize the well plate for the labs? I'm not that sure. So, okay, to standardize your experiment, I would say it takes just 1 day, just seed the cells, and then the cells will form as spheroids within 24 hours. For a cell line, if the question means how long it takes to standardize a ULA plate in the lab or in drug discovery, I think there may be 1 or 2, 3 more years, because these plates are used in preclinical studies, especially when you want to analyze the drug-induced side effects with primary hepatocytes. This is a standard to use spheroids.
Is the primary hepatocyte used in liver injury model? And are the results for any CYP3A4?
So you can use the hepatocytes in this 384 well plates, so they will also form spheroids.
Yeah, it's the primary ones used in the liver injury model. It's comparing different samples. Again, I think that's one that is, we'll look at that question and get back to you to see whether we've tested it. If the BioFloat did not interact with cells, how do the cells migrate together?
So they're forced, when they do not can interact with the surface, so normally in a normal tissue culture plate, the cells express extracellular proteins which attach to the surface, and then the cells start to adhere to this extracellular matrix. But the surface in the biofloat technology is cell repellent, and then the cells do not have the possibility to adhere to extracellular matrix, and then they decide, I would say, to adhere together. So they search the contact to other cells and start to form the network.
Mm-hmm. Since the product is cell repellent and at the same time can interact with material to produce better networks, what are the biomaterials that can actually interact with biofloat flex?
So the material, the matrix do not interact with the biofloat flex. So the biofloat flex is used to treat the surface of the 96 well plate, and the matricial is added, and the proteins in the matricial interact with the cells and not with biofloat.
And a good follow-up question to that is, what is the time required for a plate to be completely coated and ready to use?
3 minutes.
3 minutes.
Yeah.
Quick.
Yes, very quick.
It was I was thinking about the standardization. Again, looking at the data there, it is significantly quicker than alternative techniques. And plus, some alternative techniques don't actually standardize as we would define it. They are non-standard.
So comparing with other ultra-low attachment plates, we can say that our spheroids are formed very fast, and other plates need several steps, for example, centrifugation steps, and that's not needed in our plates, which is time-saving. In comparison to other methods for spheroid formation, like hanging drop, as I said in the talk, is that the hanging drop system is very, it takes a lot of time to build up the assay, and it's a complicated system.
A good question, alternative one now, is will the biofloat layer interfere in imaging, especially high resolution imaging, as this may increase the surface thickness?
No, it will not interfere. We checked that, and it will not interfere with any imaging system.
And what is the approximate thickness of the coating?
It's in the nanometer area, so there are just some nanometer.
Brilliant. Well, you've had a whole range of different types of questions there. Thank you to all those who participated in that. And again, just being sensitive to the time now. So I'd just like to say a big thanks to all the attendees for joining us. Hopefully you found what Anna Maria has talked about quite informative. And I hope it made you think about your processes and the way that you operate. More information is on the Facilitate and our Swift websites, so please have a look there. And as I mentioned at the start, if you'd like to test samples of this, then if you register your interest in the post-evaluation survey, that would be great. But for now, thank you, Anna Maria, for a great presentation. Thank you, everyone. Have a great afternoon. Bye for now.
Thank you.
Bye.